Another aspect of neuron to consider is the cytoskeleton. There are three types of long filamentous proteins within the neurons. They are located in all portions of the cell body as well as the axons. They all have a function which supports the cell and also provide specialized function. These proteins include microtubules, neurofilaments, and microfilaments. The microtubules are these cylindrical tubulin proteins involved in the axonal transport, and they occur in the axons and the dendrites, and they move material from one part of the axon to another. In this particular figure, you can see the microtubule shown as these dots. You also notice, since it's a cross-section of the axon, you are seeing these concentric rings of myelin, which we will see in more detail a little bit later. The movement across these microtubules is in two directions or bidirectional, in which there is a microtubule involved with kinesin involved in moving the material in an anterograde way, which means it's moving the material away from the cell body, and dynein on the opposite side is mediating the retrograde movement, where the material is being moved towards the body. This is an energy-consuming process, so for that reason, both kinesin and dynein are ATPase motors. The type of transport that occurs across the axon can be classified as either slow or fast. In slow transport, the material is moving at a pace of 1 to 4 millimeter per day, and that includes proteins which are soluble, includes enzymes and other cytoskeletal proteins. This rate is very similar to the rate at which peripheral nerve can repair and regenerate, which we will cover towards the end of this lecture. The other one is fast transport, which can be as fast as 400 millimeter per day. And here, the transport material are the neurotransmitters as well as mitochondria and lysosomes. The neurofilament within the neuron is depicted in this diagram, where you can see the cell body, the axon, dendrites, and then all the other processes from other cells within this picture. They look like ropes. They are assembly of cytokeratin polymers. They are involved in supporting the neuron as well as involved in the development and regeneration. In this picture, they are visible because the section has been silver stained, which is the way to visualize neurofilaments. The third type are the microfilaments. They are the thinnest type. They are made up of actin. They are involved in anchoring the molecules to the membrane, such as receptors, which form critical part of neural function. They are also involved in transferring molecules to and from the cell membrane. As actin, they are also involved in the movement of the growing axonal tip during development and regeneration. Other aspect of the neuronal structure that we should be familiarize ourselves is the synapses. And this picture depicts one such structure. Here what you're looking at is a presynaptic terminal, also referred to as bouton, of an axon, which is touching the target structure here, where it is referred to as the postsynaptic membrane. There is a physical separation between the two, and that space is referred to as the synaptic cleft. So when the impulse arrives at the bouton, the vesicles from there are released through the membrane, and the neurotransmitter is expelled into the synaptic cleft, which binds to the receptors and then internalized by the target organ or cell. These target areas where the boutons make connections are referred to as the postsynaptic densities, referring to the aggregation of these receptors. This is an EM version of the same picture, and here you can see a section of an axon where you can even see the mitochondria within it. But more importantly, what you're looking at here is the bouton of an axon with a neurotransmitter packaged in the synaptic vesicles. So this is the presynaptic axon terminal. It is making a connection with a dendrite, and you can see the density in the area where there are receptor concentration, so referred to as the postsynaptic density. And then there's a gap between the two, which is the synaptic cleft. There are different types of synapses, and you should familiarize yourself with that. They are referred to as axosomatic, in which case the axon is making a contact with the cell body. Somatic means cell. There's an axodendritic. So these are where axons are making connections with the spines on the dendrites. And then axoaxonic, where the axons are making contact with the other axons. There is a third type, which is called dendrodendritic, which is not shown there, which is basically where the dendrites are making connections with each other. There is another type of synapse which you will be learning more about in the muscle notes and labs. This is referred to as the neuromuscular junction, where it's a specialized form of synapse where the nerve are contacting the skeletal muscle, and the neurotransmitter that's used here is the acetylcholine. So we would not discuss any further because it will be covered in the muscle portion. So now let's move on to the neuroglial cells. So as we said before, in the nervous system there are two types of cells, either the neurons or the glial cells. So the glial cells, the primary role in the peripheral nervous system is to provide support and myelination. As such, there are two types of cells in this category, Schwann cells and satellite cells. This picture depicts in a diagrammatic way what these Schwann cells look like. So in this case, the green portion here and here is referred to as the Schwann cells, and then the blue portion is the axon. So these cells are making a lamellar or concentric rings wrapping around the axon. So this is the cytoplasm of the Schwann cells, and this is where the myelin is, and that's what's causing myelination. What you're also noticing here is a Schwann cell, again depicted in green with a red nucleus, just like the one that you have here in the myelinated portion. So in this case, what you're looking at is a cell which is making contact with the axons, but there is no myelination. So these are unmyelinated axons, but still supported by the Schwann cells. So how does myelination help in this nervous system? In this case, you're looking at a cell body with dendrite, axon, with myelination. And you're looking at myelination from each Schwann cell shown in this particular segment. What's happening here is that when the action potential is traveling from one part of the axon to the other, it's jumping across this gap between the two Schwann cells, which is referred to as a node of Ranvier. and we'll see the structure in a minute, what it looks like. But that is important. This jumping is important, and this myelination is also critical for the nerve conduction velocity. You will hear a lot more about that in physiology. So the presence and thickness of that myelin sheet determines the speed at which the nerve impulse propagates. Axons which have faster velocities are heavily myelinated, and conversely, unmyelinated axons have slow speeds. So larger diameter axons obviously have thicker myelin sheets and are faster at conducting the impulses. This is an EM of a Schwann cells myelinating an axon in the peripheral nervous system. And again, this is the axon here. This is the nucleus of the Schwann cell, and this is the body of the Schwann cell. And all these rings, which are the myelin, concentric rings. So this is a myelin sheath. It is important to understand how Schwann cells are actually wrapping themselves around the axons. The term that you would be asked to get familiarized with is something referred to as just Schmidt Lanterman's cleft. So how is that formed? Imagine in this picture this axon being a nice straight rope, and the rest is the Schwann cell, but Schwann cell is in a more linear, flat structure. So here you have part of the Schwann cell starting to make contact with the axon. Majority of the area within that Schwann cell is the myelin, depicted here in this blue. One part of the Schwann cell connects to the other part of the Schwann cell with these cytoplasmic processes referred to as the Schmidt Lanterman's clefts. So this part of the cell can communicate with this part of the cell. In myelination, there is a wrapping of this cell which is happening in this orientation. So the myelin is wrapping around the axon, and that creates this characteristic fishbone effect of this Schmidt Lanterman's cleft, and we'll see that in a minute how it looks like in a histological section. So here's again is a diagram which making it clear how that looks. So when you look at these pink portions here, so these are those processes of Schmidt Lanterman's clefts which are connecting one part of the cell to the other part of the cell. Majority of this blue part here, as was depicted in the previous slide, is the myelin. So as they are wrapping around, these clefts are slightly staggered, and that gives you the impression of like a fishbone. One thing also to notice here is this gap, which we refer to as the node of Ranvier. So this is the space between the two adjacent Schwann cells. Here's a histological view of the Schmidt Lanterman's cleft with a node of Ranvier. This is another view of that node of Ranvier shown here, and then this projection, which is the Schmidt Lanterman's cleft. In a unmyelinated axon, as depicted here, again you see the nucleus of a Schwann cell with multiple axons, and yet there is no myelination. So they are nurturing the axons. There's no nodes of Ranvier. Because of lack of these myelination, the speed at which the conduction is taking place is slow.